First video is here. Today we're going to be going through respiration. I'll be starting off with a little bit of basic science like what's diffusion, what is active transport, just really basic principles that I know that everyone can get to grips with how that plays in later on. Respiration comes in 4 steps. I'll be going through each one individually. They do have kind of funny words, like the first step is glycolysis, which sounds difficult, but seeing as it's the breaking of glucose, glyco means glucose, lysis equals breaking, glycolysis, it's not that bad. So yeah, I'll be running through basic science, then aerobic respiration. At the end I'll give a quick mention to anaerobic respiration, but that's really easy. And then yeah, let's crack on. Okay, so at the beginning of the video I said that I would run through some very basic principles. They are normally assumed knowledge. I've written posts on them on the basic biology if you want to read more about them. But I think it's worth quickly running through them because without understanding these, it makes understanding any stage in respiration quite difficult. So the first stage we're going to look at is diffusion. Now, diffusion is the passive movement of molecules or particles. Passive meaning it doesn't require any energy at all, they will just do it naturally, from a region where they're highly concentrated to a region where they are less concentrated or there is a low concentration. So imagine a spray of perfume, or someone farted, or someone sprayed some deodorant, or even if you put some squash in a glass of water, there's going to be a really high concentration of particles in one area where there's a much lower concentration somewhere else. This creates a concentration gradient, so you can see which way they're naturally going to want to move from this gradient. So they'll do exactly that. They will start to spread out and fill the area around them, until eventually they reach a state called dynamic equilibrium, which means the particles are equally spread throughout the entire area. So whether this one moves over here, it's okay, because that one will move over there, because they're always wanting to fill the space as much as possible and as equally as possible. But what if we're moving from a low concentration to a high? This is against the natural movement, so it requires energy. And we do this via a process called active transport. Even the name suggests, active suggests it requires energy and it's transport. So it's quite easy to remember that one. This one usually involves protein channels. So imagine we have a cell here and we have this protein channel. This is cutting off these particles getting out and these ones getting in. Now, these will naturally want to diffuse into the cell, so we need something there because there is a strong concentration gradient formed here. What will happen is this particle, let's say we're moving one of them, will move into the protein channel, and then a molecule of ATP will come and it will bind to the protein and change its shape. Now that it's changed shape, this is still cutting off anything coming out or in. But it means that this one will now go out into the area where there is a very high concentration. This has effectively moved something from a low concentration to a high. It's going against its concentration gradient. Obviously it can't stay like this forever and we need ATP, so the protein will lose its ATP and the concentration will remain locked out of the cell. So that's active transport. This next one is a little bit more chemistry than is biology, I suppose, but it's still useful to know. So this is the principle of oil rig, meaning oxidation, if something is oxidized, is the loss of electrons. So electrons are negatively charged. So if something loses an electron, it has become oxidized and it will be more positive or less negative than it was before. So if something has a charge of -1 and it is oxidized, it has lost something that is 1- and the charge will be neutral, it will be 0. Reduction is the gain of electrons. So it's a pretty handy way to remember it. Oil rig, oxidation is loss, reduction is gain. And a reducing agent will reduce something else through the process of reduction, causing it to be reduced. All of the reactions bar 1 take place in the mitochondria. Now, mitochondria are an organelle, and they exist in the cytoplasm of another cell. So here's our cell here with our nucleus. Here's a little mitochondria. Obviously, they're a lot smaller. Now, there's 4 main parts to a mitochondria. You have this place, which is the matrix. Actually, I'm going to do that the opposite way around. So this is the matrix, and this is like the cytoplasm of the cell. It is the watery goop that makes up the bulk and holds everything inside. Then we have 2 membranes. It's not like a cell with its 1 membrane here. It has 2. We have its outer membrane, which just holds everything together, and then we have this, which is its inner membrane. And they are folded into a series of projections called cristae. So it's just folded up, and then that all goes into, reaches into the middle, which is the matrix. Between the inner membrane, which is folded into cristae, and the outer membrane, which holds everything together, there is an intermembrane space. I'm just going to write it as IM space. Don't refer to it as IM, no one does. I'm just lazy and it'll be easier. So that is our mitochondria. Glucose is too big to get in, so that requires it being split, which is the first stage. And that'll take place in the cytoplasm of the cell, which is the cell's own goop. Just thought I'd mention that straight away. Now, the whole point in respiration is to produce energy, but energy isn't really a solid form, it's more of its own entity. So we need to be able to generate energy and have something to carry it. We use something called ATP, which stands for adenosine triphosphate. This said, because we have adenosine bound to a sugar, you don't really need to refer to it having a sugar, just put some H's on that. And then that is attached to 3 phosphate groups. So you may see why it is called adenosine triphosphate. Now, bonds are funny. They require energy to be made, but that by default means that they release energy when you break a bond. So when we need energy, we will take this ATP and we will break this bond here. This results in ADP being produced, which is adenosine diphosphate, because di means 2. We'll obviously then have a phosphate group just floating around, and then we have the energy that is released. Now, we can actually summarize this all into one equation, because it takes energy to make bonds, and it releases energy to break them. So if we have ATP and we take it this way, we form our ADP plus our phosphate group plus energy. But we can also take it the other way. We can provide energy to join this phosphate group to the adenine diphosphate and reproduce ATP. And this is the basis of respiration. In school, and I'm talking primary school, we are taught respiration as one very simple equation. And to an extent, this is actually true. So we have C6H12O6. That is glucose. I'm not going to draw the groups on. Then we join that with 6 molecules of oxygen. We all know we need oxygen to breathe, which is why we should stop chopping down the trees. And that produces 6 molecules of carbon dioxide plus 6 molecules of water. And then we can also think about the energy that we produce. It's important to think of this equation because it summarizes the whole process. Now, we already know that aerobic respiration, with oxygen, comes in 4 steps. But it is useful to think of this because we know we need oxygen, we know that we produce carbon dioxide, and we know we produce water. In going through with this, we're just going to find out how. Going into the first step, we're thinking about glycolysis. I know it sounds sciencey, so once I explain it, you can talk to people and pretend you're smart. Glycolysis is simply the breaking of glucose. So glyco equals glucose, and lysis, if you ever see the term lysis, it means to break. So hydrolysis is to break a bond by addition of water, Osmotic lysis is to break something through osmosis. Lysis means break. So what we do is we start with our glucose, and then we split our glucose into 2 molecules of pyruvate. Now, a little tip to remember this is glucose, C6H12, is a 6-carbon molecule. We are splitting that into 2 molecules of pyruvate, and that is a 3-carbon molecule. So, simple maths. To start off this splitting, we require an investment of 2 ATP. And this will kickstart everything. This is what's going to get the breaking to start. But by the end of it, we actually produce 4 ATP. Taking out the investment phase, that means we generate 2 ATP for each cycle of glycolysis. Now, that's a tiny amount of energy, so we don't really use glycolysis for energy normally. There is, the circumstance of anaerobic respiration, and I'll explain that later, but right now we can just kind of disregard that. What we can't disregard is the fact that it also produces this precious molecule, NADH, and it produces 2 of them because there's 2 molecules of pyruvate. And what happens is we have 2 molecules of NAD+, it swoops in, and it becomes reduced because it's gained this proton. It takes 2 hydrogens, and it forms 2 molecules of NADH. This goes on to the final step, so right now we can disregard it. What we need to think about is pyruvate going on to the next step. Now, the link reaction is often not regarded as its own step, but I was taught it as both its own step and as its separate step by a different biology tutor, and I always found it much easier to remember when it was its own step. Now, one of the reasons we do glycolysis is because glucose is far too big to enter the cell by itself. So when we split it and form pyruvate, it breaks it down and it can start to enter the mitochondria. So between the cytoplasm of the cell and the matrix, this reaction takes place. And what we do is we take our pyruvate. Remember, this is a 3-carbon molecule. And then we take some coenzyme A. You don't need to know much more about it apart from the fact that it's called coenzyme A. And another molecule of NAD+. We will then react this and we will form acetyl coenzyme A, some NADH from that NAD we had before, it's nicked a hydrogen, and some CO2. So the NADH, that's going to go on to the last step. We don't need to think about that right now. The CO2 is going to be exhaled. But just to note, all of a sudden we've got CO2, which means something has got to have lost a carbon. That was our pyruvate. While it is acylating this coenzyme A to form acetyl coenzyme A, it's a 2-carbon molecule. So it's lost a carbon, and that's exhaled as CO2. So we know where that's going, we know where that's going. Let's think about where this is going. Our acetyl coenzyme A will now be in the matrix of the mitochondria, and that will go into something called the Krebs cycle. I'm just going to write Neo because you're in the matrix. Okay, that was a terrible joke. Okay, so we have the Krebs cycle. We're going to start with our acetyl coenzyme A. Now remember, that's a 2-carbon molecule. Already here is a 4-carbon molecule. That will come along and it will join with our acetyl coenzyme A to form, hey presto, a 6-carbon molecule. We don't need to know the names of it. If you'd like to know more about this step, you can read my degree level post, but to be honest, you don't really need to know more than this unless you're studying biochemistry or some other similar degree. This is all you really need to know. So this 6-carbon molecule will then cycle back to the 4-carbon molecule, meaning it's lost 2 carbons, and it loses them in the form of CO2. Oh, and we know where that's going. We also generate a molecule of ATP, But again, this is a pretty negligible amount. The Krebs cycle is quite a slow cycle, so we don't tend to use it for energy. It only makes one. What it does make, which is the very important bit, is FADH2 and 3 molecules of NADH. These do pretty much the same thing, and we've now got 4 molecules of it. So we generated these. This will continue to cycle. We will have 4 carbon join with acetyl coenzyme A to form 6 carbon. That'll lose 2 carbon to carbon dioxide, cycle back to 4, and it'll constantly just repeat. And every single turn we get these. They're going to go on to the last step, which we're about to get onto now. This one has a very long name, but the name is actually very simple, so whatever you do, if you're trying to convince someone you're smart, just don't explain it to them, because it really is very simple. This is the step of oxidative phosphorylation, often misconceived as the electron transport chain. If you're a student, like doing A-level biology or so, you will probably have heard of this being referred to as the electron transport chain. It does involve it. The electron transport chain is part of it, but it is not the name of it. It is oxidative phosphorylation. So think about what we're doing. The whole point in respiration is to make ATP. And to do that, we are phosphorylating ATP. So when we phosphorylate, we're adding phosphate to something else, and that will make our ATP. So that explains half the name. Now, we also know from our primary school equation that we need oxygen. So this is happening in the presence of oxygen. Oxidative phosphorylation, the addition of phosphate in the presence of oxygen. Now, I mentioned the mitochondria and how it is folded into these finger-like projections called cristae. Oh, that's better. So we're going to take one of our cristae here. Inside the cristae are little proteins, and they are embedded onto the surface. And then we have this over here, and this is called ATP synthase. I'll give you 3 guesses as what that does. Yeah, synthesizes ATP. We'll get onto that later. So I've been banging on about NADH and FADH2, seeing how important they are for a while now. Well, let's find out why. These are called proton carriers because they carry these hydrogen ions. What happens is they split to form NAD and FAD, and then they will go back up to the other 3 steps, and they will carry on to be reduced and take the hydrogens away, because if they don't do that, those steps can't continue. You need to have these there. It also generates electrons and protons, each of which will go their own way. So let's think about our electrons. It will go into this protein here, and it will reduce it, because reduction is gain of electrons. It will then leave that, causing it to be oxidized, and it will reduce this protein. It will then leave this protein and go on to the next one that is now oxidized, and this is reduced. And this series of reduction and oxidation is just called redox. They are redox reactions. As it keeps jumping, it produces energy. Remember how I talked about active transport before? This is the movement of something from where it is more lowly concentrated or in a lower concentration to an area where it is more highly concentrated. So in this intermembrane space, there are loads of hydrogen ions already there, which means these hydrogens from the NADH and FADH2 can't enter by diffusion because it's just against the concentration gradient. What will happen is this energy will be used for active transport, meaning that the protons can enter. Once inside, they cannot leave through this membrane.
it's airtight, you know, it's a membrane, it's what it's for. So the only way out is through this protein here, ATP synthase. Now obviously there's a lot more in here than there are there, so you don't need any energy for this, they just diffuse. When these protons leave this channel, this is the important bit, they produce energy. And it is this energy that is used to get our ADP plus P to make our ATP. We are harnessing the energy of the protons leaving through this protein channel to make our ATP. So much so that for each glucose molecule, we actually generate around 32 ATP. Seeing as starch is just a massive line of glucose, you can see how much energy we are actually able to produce. Obviously, we can't have loads of electrons and protons floating about. I mean, we've fulfilled most of the equation. We've got our CO2 being produced from the other steps. We have our glucose being used because it's having these proton carriers being reduced. And then we've also produced, so we've fulfilled most of our primary school equation. We've used glucose, we have produced carbon dioxide, but we haven't thought about why we need oxygen. That hasn't come into the other stages yet, and we haven't thought about where we're getting water. So what will happen, this is aerobic respiration, meaning it needs oxygen. So imagine we start with O2, that's going to split to form 2 molecules of oxygen. Oxygen is referred to as the final electron acceptor because it does exactly that. It will take this electron down here and it will go to this oxygen. Because if this electron stays here, then this electron can't come from that protein to this one, which means that one can't go to there, which means one from here can't go to there, which means it completely stops it. It means no energy is being produced for active transport, and then it means nothing is leaving here because there aren't any proteins being built up in the intermembrane space to leave to provide the energy for this ATP, which is why we need oxygen. We also have these protons here, and they will also join with that, and that will form water. In fact, for each glucose, we get 6 molecules of water, and that will be exhaled. We have fulfilled our primary school equation. So, just to run through aerobic respiration, we have glycolysis, and that's glucose to pyruvate. And then that also generates NADH. That'll go to the last step. We have the link reaction, which is pyruvate, which forms the important acetyl coenzyme A, plus more NADH, which goes to the last step. That just obviously, that's all that matters. Third, we have the Krebs cycle, and that is the acetyl coenzyme A goes into the cycle, and out of that cycle comes 3 molecules of NADH and a molecule of FADH2. Now our electron transport chain, which if you think of it systematically is actually very, very simple. We get our protons and our electrons coming from all of these. They will all do this. The E- go through the redox reactions. That produces energy. That's for the active transport of protons. They then diffuse through the ATP synthase, because that is the only way out. That energy is then used for our equation that we mentioned at the beginning, ADP plus phosphate plus energy, to make ATP. And then we have our oxygen plus our electrons plus our protons to make H2O. Oh, I've drawn it down there. There, that's better. No one ever tell. To get our H2O. So we've used oxygen, we've produced our carbon dioxide, we've produced our water, and we've used our glucose. And that is why we need respiration. Now, what I didn't mention before, which I was supposed to but forgot, is this acetyl coenzyme A that's used in the Krebs cycle. I'll just draw the top of it here. This doesn't have to just be made from glucose. We can actually use fatty acids. Now, in the lipid structure post, I said about how lipids in the form of fat, which we store as triglycerides, contain fatty acids. So we can convert fatty acids into acetyl coenzyme A, which removes glycolysis. It means that when we don't have enough glucose, we don't need that because we can just use stored fat and push it in. The problem is this isn't as efficient, and we are built to run off glucose, and our brain runs on glucose. The cells in the brain respire using glucose. So although you can limit the amount of glucose you intake to use fatty acids in the form of stored fat instead, to produce this acetyl coenzyme A, if you limit it too much, you can end up using amino acids from proteins. So be careful on that one.
Okay, so that's aerobic respiration done. If you found it useful, make sure to like it below and subscribe. And if you'd like to know any more or have something else to help learn, there is a corresponding blog post on respiration, which is linked at the bottom, just down there. Go have a look. If you're a degree level student, then there's a link below to the degree level posts on each stage, but they are a lot more advanced. I wouldn't recommend it if you're just looking for someone who's doing A levels or something similar, or you're someone who is a personal trainer or someone who's a bit of a nerd or any other reason, because you don't really need to know more depth than that. Make sure to go over to Facebook, again it's just Know How Nutrition, or on Twitter at Know How Nutri. Give a like or a follow or anything else. To be honest, it'll really help the page out and get it out there and just show how easy this kind of stuff really is. And I know that it was a long video and it may take a bit longer to get, but once you get it, you can apply it so many more things, such as how fatty acids can be converted to acetyl coenzyme A, and that is how basically the basis of all calorie restricted diets. So it's a lot easier once you actually know it and you know how to restrict your own calories than it is to do it just because someone told you to and asked for money to do it.